So today we'll be talking about DNA replication. Before we get into the process of DNA replication, we will look at what are all the things that are necessary for DNA replication to occur. So first things first, we will be requiring substrates. Now substrates for DNA replication, of course, would be DNTPs, which would be DATP, that is deoxyadenosine triphosphate, deoxyguanosine triphosphate, deoxycytidine triphosphate, and deoxythymidine triphosphate. Although we will be using the triphosphate forms of the nucleotides, the nucleotide which eventually will be put into the DNA strand will be a monophosphate. Okay, the two other phosphate will get removed and a monophosphate form of the nucleotide will be attached to the growing DNA strand. Another requirement would of course be the template DNA. Template DNA is the original DNA which will be used as a template to synthesize the new DNA strand. Primers, which are short RNA strands. These are very short RNA strands which will be around 10 to 30 base pairs in length, and they will be synthesized by the primase enzyme. And these are necessary because DNA polymerase cannot start off polymerization reaction, which is adding in different nucleotides to the growing chain, that is the new synthesized chain of the DNA, without having a 3' OH group. So it needs a free 3' OH group, and without this DNA polymerase cannot start adding new nucleotides. And so this 3' OH group, the free 3' OH group, is provided by the primer. So once a short strand of primer has been synthesized, its 3' end will have a free OH group to which a 5' phosphate group can be attached, and the chain elongation will take place further and further and further. So this phosphate group will be attached to a 3' OH group, which will be attached to a 5' phosphate, and then the chain elongation will continue in that way. The most important thing for a DNA replication would be the enzymes that will be used. Now we'll be looking at each and every enzyme individually. Helicase is the first enzyme that comes into use. Helicase is a DNA unwinding enzyme. It's going to break down the hydrogen bonds that are formed between the nucleobases. So adenine forms 2 hydrogen bonds with thymine. So adenine is going to have 2 hydrogen bonds with thymine, and guanosine is going to have 3 hydrogen bonds with cytosine. Okay, so helicase is going to break these bonds, and therefore the 2 DNA strands are going to become single strands. Okay, so it's going to do the unwinding. Another enzyme is the single strand binding proteins, or as they are known as SSBs. Now single strand binding proteins are, like the name suggests, are going to bind to the single strand. So once helicase has broken down the hydrogen bonds, you will have 2 single strands of DNA molecules. Now these single strands can again come back together and form a double strand by forming a bond between the adenine and thymine, or guanine and cytosine. To stop this from happening, single strand binding proteins, which are basically proteins, are going to bind to both the single strands, and because of these proteins binding to the free single strands, they cannot come together again and form the hydrogen bonds. The other enzyme that is found in DNA replication is topoisomerase, which is a class of enzymes which were first discovered by James C. Wong. Topoisomerases go before helicases. So for the process of DNA replication, topoisomerases are the first enzymes to act. So they are going to remove the supercoils which is there on the DNA. So DNA generally is in a packaged form. It is coiled on itself. So topoisomerases are going to remove those coils, and then helicase is going to unwind the 2 strands of the DNA. Now after helicase starts unwinding the 2 strands of the DNA, because of the unwinding of the DNA itself, because DNA strand is also helically twisted on itself, even a single strand of of DNA is helically twisted on itself. So when you are going to unzip the DNA, remove the hydrogen bonds, there is going to be stress which is going to be formed further up on the DNA strand, and that is going to be removed by DNA gyrase or topoisomerase 2. Now DNA gyrase is one of the topoisomers that are found in the entire class of topoisomers, but it has a very specific function that it goes after the helicase activity. So before helicase is topoisomers in general, they are going to remove the supercoils. Then comes helicase, which is going to unwind the DNA. Then comes DNA gyrase or topoisomerase 2, which is going to remove the stress and coils which are going to be formed because of the helicase activity. Okay. After that comes the DNA clamp or the sliding clamp enzyme. It's actually a part of the DNA polymerase 3 holo enzyme. So DNA polymerase 3 is made up of multiple subunits, and one of those subunits is the DNA clamp or the sliding clamp enzyme. Now the function of the DNA clamp or sliding clamp is that, like the name suggests, it's a clamp. So it is going to hold on or latch on to the single strand DNA and make sure that the DNA polymerase is not going to detach from the single strand DNA. So once the DNA polymerase attaches to the single strand of DNA and starts synthesizing a new strand of DNA, DNA clamp is going to make sure that it does not dissociate from it. So it attaches it to the template strand. Okay. The next enzyme would be primase. Like I was saying before, primase is an enzyme which is going to add a primer, which is going to be a short length RNA. Okay. You always need to remember this, that the primer is going to add an RNA segment and it's not going to be a DNA segment. So it's going to be a short RNA segment which is going to provide with a 3' OH group. So it's going to provide a 3' free OH group, using which the DNA polymerase can synthesize the new strand of DNA. DNA ligase is another enzyme that is used. This enzyme is going to attach the two strands of DNA and also the Okazaki fragments to each other. Okay. So DNA ligase is going to attach the two strands of DNA and also the Okazaki fragments. Another enzyme is the telomerase enzyme. Now this is an enzyme that is found only in eukaryotes. It's not found in prokaryotes, because only eukaryotes have different portions in the chromosome, like the telomere section of the chromosome. Now telomers are going to lengthen the telomeric DNA, and they do this by attaching repetitive sequences of DNA to the telomere region. Okay, whenever replication of eukaryotic DNA is taking place, telomere enzymes are going to add random repetitive sequences to the telomere region of the DNA, thereby lengthening the telomeric DNA. Okay, that's the main function of telomerases. Next we move on to DNA polymerase. This is of course the most important enzyme that is there for DNA replication process, because it is the polymerase. Polymerase is the enzyme that is going to be adding the new nucleotides to the growing strand of the DNA. It's also going to be doing proofreading. It's also going to repair if there are going to be any damaged DNA particles in the new strand of DNA that is being synthesized. Now, based on whether it's found in prokaryotes or eukaryotes, there are different kinds of polymerase enzymes that are found. Now, in case of prokaryotes, there are 5 different kinds of polymerase enzymes that you will find, named as DNA polymerase 1, DNA polymerase 2, DNA polymerase 3, polymerase 4, polymerase 5. Okay. Moving on, DNA polymerase 1 is the most abundant polymerase enzyme that you will find in prokaryotes. Close to 95% of a cell's polymerases, the DNA polymerases, are usually DNA polymerase 1. It has both 3' to 5' and 5' to 3' exonuclease activity. Okay, so both 3' to 5' and 5' to 3' exonuclease activity, and because of this reason it can do excision repair in both directions. Excision repair meaning that if there has been a wrong nucleotide that has been added to the growing chain of the DNA, the new chain that has been synthesized just now, a new different nucleotide has been added, or a wrong nucleotide has been added. That needs to be removed and the correct one needs to be added. Now for that, the removal can occur either from the 3' end to 5' end or from the 5' end to the 3' end. Not all polymerases have the ability to do exonuclease activity from 3' to 5' and 5' to 3'. It's only polymerase 1 that has both 3' to 5' and 5' to 3' exonuclease activity, so it can take part in excision repair. It also processes Okazaki fragments, meaning that it is going to remove the primers which are there in the lagging strand. It's going to remove them and then replace the primers which are RNA nucleotides with DNA nucleotides. So it's going to be processing the Okazaki fragments. It adds nucleotides extremely slowly, so it adds around 15 to 20 nucleotides bases per second. So this addition is going to be per second. So this is extremely slow. So once a primer has been synthesized by primase, DNA polymerase 1 will start off the synthesis of the new strand. But after a after a certain period of time, it will be polymerase 3 which will take over. Polymerase 3 is the major enzyme for DNA polymerase reaction. Polymerase 1 basically does excision repair and processing of the Okazaki fragments, because its addition of new nucleotides is very, very slow. But it is the one that starts off the replication from the primer. So from the primer, starting the replication is started by polymerase 1, but afterwards it's taken over by polymerase 3. The second enzyme is polymerase 2. This one has only 3' to 5' exonuclease activity, so it can do excision repair only in the 3' to 5' direction. It plays a very important role in bypassing lesions. So lesions are those parts of the DNA which are erroneous, meaning that there is an error in the template DNA itself. Maybe two thymine nucleotides are forming a dimer, or there has been a mutation or some such has happened. So there is a error in the template DNA, and therefore when the polymerase is reading the template DNA and adding the new nucleotides for the growing strand of the DNA, if it comes across a lesion, meaning an error, it's going to stop replication. Okay, so usually the major enzyme, which is polymerase 3, which will be doing the polymerase activity, if it comes across an error, it stops the polymerase reaction and it will not continue further unless and until that error is removed. Now because during replication the error cannot be removed from the template strand, polymerase 2 will come and it will skip that error part. Now this is not good for the DNA as a whole, because it will be inducing mutations wherein you are taking out a part of the DNA from the template DNA and also from the newly synthesized DNA. But this is the way cells deal with errors or mutations if they are occurring during the replication process. Okay, so that's the point with polymerase 2. Its major function is to bypass lesions. Okay, polymerase 3 is the main enzyme or the major enzyme for DNA replication process. So initially polymerase 1 will be the one that will start, but polymerase 3 is the one that's going to take over and complete the DNA replication process. So polymerase 3 is the main one that has the polymerase action on it. Also, it has proofreading action on it. Proofreading meaning that as the nucleotides are being added, the polymerase is going to read through the new nucleotides which have been added to the newly synthesized strand and make sure that the correct ones have been added. So proofreading is figuring out whether or not there has been an error in the polymerase reaction. Okay. Polymerase 4 is known as an error-prone polymerase. It brings in non-targeted mutagenesis because it also does translation synthesis. So like polymerase 2, If polymerase 4 comes across a error in the template strand, it is not going to repair the template strand. It will simply add in random nucleotides, or it will skip that part of the template DNA completely. It will not replicate that part, it will skip that part completely, or it will randomly put in different nucleotides. Okay, so that is the reason why non-targeted mutagenesis is something that will occur in polymerase 4, in polymerase 5 and in polymerase 2. So polymerase 2, polymerase 4 and polymerase 5, because all three of them undergo translation synthesis, meaning that whenever they come across an error during the replication process, they are either going to skip that part of DNA or simply going to add random nucleotides. So this is going to create a mutation, and all three of them are responsible for non-targeted mutagenesis. Okay. Polymerase 5 of course is also error prone like polymerase 4, and like I was saying, it also does translation synthesis, meaning that whenever it comes across an error, it's either going to skip it or going to add random nucleotides. On to the polymerases, the DNA polymerases which are found in eukaryotes. You have different kinds. The major ones I will be talking about here: polymerase alpha, beta, delta, lambda, epsilon, mu, gamma and theta. Okay, we will go on with each and every one. Polymerase alpha, it lacks 3' exonuclease activity and it initiates replication. So polymerase alpha is the one that is going to initiate replication whenever DNA replication starts. Also, it lacks 3' to 5' exonuclease activity. Moving on to polymerase beta, its major function in eukaryotic cells is that it can do base excision repair, meaning that it can remove just one single base from the growing strand, the newly synthesized DNA strand. It can remove one single base, which is erroneous, which is not supposed to be there and has been put there. So it can remove that one single base and replace it with the correct base. So that is known as base excision repair, when you are just taking out one single base which is wrong and then putting in the correct base. So polymerase beta, its main function is to do base excision repair. Polymerase delta can do both replication and repair. So its major functions would be replication, that is the polymerase action, and it can also repair if there are any mutations occurring. Polymerase delta, it can do double strand break repair, meaning that in the synthesized strand of DNA, if there is a double strand break, meaning that now here is a double-stranded DNA and a break has occurred, meaning that there are some nucleotides which are missing there. Polymerase delta is going to simply join those two strands just like that by themselves, or it is going to put in new nucleotides. It's randomly going to put in new nucleotides and then do end-to-end joining of a double strand break. So polymerase delta can do a double strand DNA break repair. It can also do base excision repair, but only when polymerase beta is not functioning. So if polymerase beta is not functioning, then polymerase delta is going to take up base excision repair. It also resynthesizes missing nucleotides, like when there is double strand DNA break repair occurring. So if a double stranded DNA break has occurred, the repair can be done by simply joining those two DNA strands together, or polymerase delta is going to put in random nucleotides. So it's going to resynthesize the missing nucleotides. It does not have a template to go on. It is going to synthesize for the template strand and to the new strand. So it's going to be adding new nucleotides just like that, randomly. Okay, so this will lead to mutations. Polymerase epsilon, it can do only leading strand synthesis, Okay, so polymerase epsilon can only do leading strand polymerase action, meaning it's going to add nucleotides onto the leading strand only. It cannot add nucleotides onto the lagging strand. That will be taken up by the other polymerases. Polymerase epsilon can also do base excision repair, but again only when polymerase beta is not present. When polymerase beta is not present, the other polymerases will take up base excision repair, but if polymerase beta is present, then it is going to do base excision repair. Then you have polymerase mu, which is going to be again resynthesizing missing or damaged nucleotides. This is known as non-homologous end joining. NHEG stands for non-homologous end joining, and this is something that is done by polymerase mu, wherein it's going to synthesize missing or damaged nucleotides. So damaged nucleotides meaning that something like a thymine dimer is formed, meaning that two thymine nucleotides which are right next to each other are going to form bond between them. Instead of one thymine forming a bond with another adenine on another strand, it's going to form a bond with a thymine right next to it on the same strand. Okay, so those are damaged nucleotides. So polymerase mu can do damaged nucleotide repair, meaning that it's going to either remove those two thymine nucleotides completely from the newly synthesized strand, or it's going to put in random nucleotides into it, or it's going to resynthesize missing nucleotides like the same thing that polymerase delta does, polymerase lambda does, wherein it's simply going to add in random nucleotides. So all of these are going to end up in creating mutations in the DNA. Next we have polymerase gamma and polymerase theta. Both of them can do replication, meaning that they have polymerase activity. They can repair if there is any damage that is occurring in the newly synthesized strand, they can repair it, or if there is a damage already occurred in the template strand, it can repair that. It has proofreading activity, meaning that the newly synthesized strand is going to be read by polymerase gamma or by polymerase theta, and it's going to check whether or not the newly synthesized strand has the same basis complementary to the template strand or not. And if it is not complementary to the template strand, the wrong ones are going to be removed and repaired. Also, it has 3' to 5' exonuclease activity, meaning that it can remove a stretch of DNA from the 3' end to the 5' end, but it cannot do the opposite, meaning it can't do exonuclease activity from 5' to 3' end. So these are the different DNA polymerases which are found in the eukaryotic cells. Now that we know what all are necessary for the DNA replication process, we can move on to the actual steps in DNA replication process. The first step of DNA replication process would be the ori site, or the origin of replication site. In case of prokaryotes, there is only one origin of replication found on the entire DNA. In case of eukaryotes, there are multiple ori sites which are found on the chromosome of eukaryotes. Now, ori sites are usually rich in adenine and thymine bases. Because adenine and thymine bases have just 2 hydrogen bonds between them, it's easier to break it rather than a guanine and cytosine rich region, in which case there will be 3 bonds between guanine and cytosine, and it's much more difficult to break them. So ori sites usually are rich in adenine and thymine molecules, and they are usually anywhere between 10 to 250 base pairs in length. Now, ori sites are the sites at which proteins called as initiator proteins are going to come and bind, and these are going to be a sort of a beacon or a signal for the other proteins, the other enzymes which are involved in DNA replication to come and bind and then start the process of replication. So initiator proteins, like the name suggests, are initiators for the process of DNA replication. They are going to be binding to the ori site. Now, an ori site in case of E. coli is DNA So in case of E. coli, the ORI site is known as DNA A. So this is the ORI site for E. coli. The next step in DNA replication will be, of course, the unwinding of the DNA strand, and there are three major enzymes which are involved in it. Topoisomerases, like I was explaining, is going to remove the supercoils. After comes the helicase, which is going to break down the hydrogen bonds between the two strands of the DNA, which is going to give you two single strand DNA, and so it's going to form a Y junction, meaning that you will have double stranded DNA here, and here will be your helicase enzyme, and wherever it has unwinded, you're going to have the unwinded single strand DNA, and so this place is known as the Y junction. And after helicase comes DNA gyrase or topoisomerase 2, which is going to remove the coils and strain that is put on the double stranded DNA because of the unwinding which is caused by helicase enzyme. Okay, once the unwinding has occurred, you will have formation of primers. Formation of primers is going to be carried out by the primase enzyme, and primase enzyme is going to add short length RNA sequences. So it's going to be ribonucleotides and not deoxyribonucleotides which will be added, and those will be complementary to the single strand template DNA. So there will be a primer which will be added, a short length ribonucleotide, and that will be complementary to the template DNA strand, and this is going to provide the DNA polymerase with a free 3' OH group from which the replication can start. The first to start replication in case of prokaryotes will be polymerase 1. So Pol 1 is going to start off the replication process, but it will be Pol 3 which will take over the replication process from then onwards. So for a short length, it will be polymerase 1. In case of prokaryotes, it will be polymerase 1 which will be starting off the replication process, then polymerase 3 which will continue the replication process further on. Now polymerases have only 5' to 3' polymerase activity, meaning that they can add nucleotides only from the 5' end to the 3' end. So this will lead us with a lagging strand and a leading strand. So a leading strand is going to be the strand which will have polymerase activity from the 5' to the 3' end. So the polymerase activity is continuing in a continuous manner because it's from the 5' to 3' end. There is another strand, the opposite strand, in which the polymerase activity should happen from the 3' to 5' end, and because polymerase cannot have 3' to 5' polymerase activity, it cannot synthesize in this direction. This direction synthesis cannot occur, and therefore there will be multiple primers which will be synthesized at different intervals, and the space between them will be filled up either by polymerase 1 or polymerase 3. So here the primer has been synthesized by primase at different intervals, and then the space between them will be filled up with polymerase 1, which is going to fill up the space between the two different primers, and this synthesis will of course occur in the 5' to 3' direction. Okay, so this would be called as the leading strand, and this one would be known as the lagging strand, and these small pieces of DNA which are filled in between the primers are known as the Okazaki fragments. Okay. the next step would be after the entirety of the DNA has been replicated. So once the leading strand and the lagging strand both have been replicated completely, the next thing would be removal of primers and gap filling, which will be done by polymerase 1. So as the replication is continuing and going on, it is moving forward, the primers which were there on the leading strand will be only one. In case of the lagging strand, there will be multiple primers. So those will be removed and they will be replaced by deoxynucleotides, and that will be done by polymerase 1. After that, the step would be ligase, wherein ligase is going to form the bond between the two single strand DNA, and it's also going to be forming the bond between the free phosphate group and the free OH group in between the newly synthesized. Next enzyme will be ligase, and ligase is going to be ligating the two single-stranded DNA molecules. So you have the template strand and the newly synthesized strand, and the nucleotides which are there in between each of them will be ligated by the enzyme ligase. Also, whenever the primer is removed and a new deoxynucleotide inserted sequence has been synthesized, so if you think of the leading strand, you have a primer and then the DNA which has been synthesized. Now this primer has been removed using polymerase 1, and then a new strand which will be having deoxynucleotide has been put in. Now if you think of the lagging strand, you have multiple primers which are there, and these also will be removed by polymerase 1 and they will be replaced by deoxynucleotides. Of course, you have in between them already synthesized DNA fragments. Now these two strands are going to have the backbone of sugar and phosphate, like any DNA strand. Now between the sugar and phosphate backbone of these two, there will be a gap, because the 5' phosphate is not going to be bonded with the 3' hydroxyl group of the sugar molecule. So the 5' phosphate and the 3' hydroxyl group will not be bonded between these two, and that also, the bonding also will be done by DNA ligase. So DNA ligase does two things. One, it is going to bond two of the single strands of the DNA together, making it a double-stranded molecule. It is also going to form the bond between the DNA backbone, wherein you have a sugar molecule and a phosphate molecule. The 5' phosphate molecule of one is not going to be bonded with the 3' hydroxyl group of the sugar of another, and this gap in the backbone is going to be filled up by DNA ligase again. The last step, of course, will be the termination of the DNA replication, and this occurs whenever the polymerase comes across something called as the TER sequence. TER sequence is also the termination of replication sequence. So TER stands for termination of replication, and this is a sequence in the DNA where the polymerase is supposed to stop replicating. Now how it happens is that whenever there is a TER sequence in the DNA, TER binding proteins are going to bind to the DNA. So if you think of a DNA strand like this, and then you have a TER site here, okay, so this will be your TER site, and to which TER binding proteins are going to be bound. Now because TER binding proteins are bound to it, the DNA polymerase cannot move forward. So it's going to be stopped physically because the DNA strand is filled up with TER binding proteins, and that is where the polymerase stops, and that is when the termination of DNA polymerase takes place. After that, is proofreading. Proofreading is when the newly synthesized strand of the DNA is going to be read, or rather checked, for any abnormalities. If there has been a nucleotide missing, or if a wrong nucleotide has been put in, or if there has been a break in the newly synthesized strand, all of these things are repaired, and that is the reason why proofreading happens. So proofreading will be the final step of the DNA replication process. So you will be starting off with oocyte, then unwinding, then formation of primers, which leads to continuous and discontinuous replication, forming Okazaki fragments. Then you will have the removal of primers, and the gap wherever the primer was there will be filled up by DNA polymerase 1. Then comes ligase, which is going to ligate the two strands of the DNA, and it's also going to ligate the backbone wherever there is a 5' OH and a 3' hydroxyl group on the deoxyribose sugar. Then comes termination, which wherever the ter sequence is there, ter binding proteins will be there, and they will physically stop the DNA polymerase from moving forward. The last step would be proofreading, and proofreading is going to check for any abnormalities, mutations, or missing nucleotides, and it is going to repair it, and this ends DNA replication. Now, because prokaryotic DNA has just one oocyte, one origin of replication, it's going to start from one end and then finish at the other end. Because eukaryotic replication, eukaryotic chromosomes have multiple oocytes, they are going to start off replication simultaneously in all of the oocytes. So when you look at a eukaryotic cell which is undergoing replication, it will look like there are multiple bubbles which are there inside of the DNA strand. This is because everywhere there is a oocyte, the DNA is going to get unwinded. And because the unwinding is happening at multiple sites at the same time, wherever the DNA has unwound, it is looking like a bubble or like a eye. So this is known as the replication bubble or the replication eye in case of eukaryotes. In case of prokaryotes, because there is only one origin of replication, it is going to start from there only and then end off in the other end. And because of this reason, in case of prokaryotes, it's going to form the Y junction. It does not have the replication eye or the replication bubble.